Mr. B here. This is a slideshow on operons. And the operons, understanding them really are key to understanding the GFP lab. So here we go. First, I want to start out with just a quick reminder about transcription. Transcription is how we take DNA and we copy a strand of DNA to make RNA, and then the RNA, that's called a transcription. Transcription is making that RNA, single strand, and then remember, translation is taking RNA and making it into protein. One point is that with prokaryotes, which is what we're basically going to be talking about here in this video, prokaryotes go from transcription to translation in one step. And I talked about that in the past, so I'm not going to revisit that, but just remember that RNA polymerase is the enzyme that helps copy that strand of RNA. Okay, so operons. What's an operon? An operon is a method to control gene expression, and this is in prokaryotes only. Okay, big thing to know. And what are operons? Well, they're groups of genes that ultimately produce proteins. Of course, well, genes produce proteins. But these operons, there's two types of genes in operons, and so they have these structural genes, and then they have regulator genes. And all these visuals that are coming up come from this website right here, which is pretty good. And notice SUNY.edu. So, you know, those educational, and generally you can get some pretty good stuff from educational websites. And so here is this visual here. This is a visual of an operon. The operon would be the green stuff. The green stuff is representing a chunk of DNA, and it's not shown real well, but it's, you know, DNA is double-stranded, or I'm sorry, a double helix. And on an operon, it has a regulator, a promoter, and an operator, and then these genes here, okay? And so the structural genes are these guys right here. These are the genes that are going to produce proteins, like, for example, taking lactose and breaking it down into glucose. Sound familiar? Remember cellular respiration lab? So one thing to note is that, again, with this prokaryote, that a single, this would ultimately, you know, would, if we can have the polymerase, the RNA polymerase go along here and make the mRNA, what you would make is mRNA that can produce multiple proteins. And so, generically speaking, you can make protein A, protein B, and protein C, all with one single mRNA molecule. Phew, I can't say that. All right, and so those groups code for different enzymes right there. Okay, the regulator part. Remember, we talked about there was the structural genes. Now we're going to talk about the regulator genes. And what these do is, hey, they produce proteins that regulate genes, and it's like kind of circular in nature, isn't it? Yeah, well, that's why we got visuals here. Okay, so what you're seeing here, this is a visual of E. coli's lac operon, and this lac A, lac B, lac C, those are genes that would produce enzymes which help E. coli break down lactose into glucose so that it can use it for energy, for respiration. So what you see here, now a regulator gene, that would be right here. So we could have RNA polymerase go ahead and read that chunk of DNA right here, and what it would do is it would ultimately make mRNA, which would then ultimately make a protein, which is going to be called the repressor protein. So right from here, it's going to make a repressor. And when lots of glucose is present, E. coli would much prefer to basically use the glucose and not use lactose. So what it would do is this regulator gene would be expressed. It would make a protein, and that protein would pop right here. Okay? So as long as this protein is hanging out on that chunk of DNA, the RNA polymerase physically will not be able to go cranking along the DNA, and split the DNA up and make the copy, or, well, I shouldn't say that, split the DNA, but the polymerase won't be able to make the copies of one strand of the DNA to make these proteins to metabolize lactose because, hey, I don't need to. I've got glucose. So the lac operon, you think, okay, E. coli would like to use glucose, but what if lactose was the only sugar present? So let's flip it around. Let's say, well, what if we just gave E. coli lactose? Would the E. coli die? Well, what will happen is there's an isomer of lactose that's found when lactose is present, and it has a particular shape. It's called, and this is called allactose. I might be saying it wrong. I don't know. But its shape is peculiar in that what it will do is it will fit into the repressor protein. Okay, so when that fits into that repressor protein, what it will do is it will pull that repressor protein off. And that will allow the RNA polymerase to go to the promoter site, which it will have the start code on there, and it will start cranking along, and it will make the proteins that will help E. coli break down lactose into glucose. Pardon me. And so what this does is now, because we've taken the repressor protein off, that allows for transcription. Okay, now, here's a new word, inducible operon. Okay, inducible, what does that mean, yo? Well, an operon consists, here's operon, it consists of a promoter, an operator, and the genes. Okay, now, and what I've got here, this is kind of circular. Well, this repressor protein is produced by the regulator gene, right? And so the lac operon is called inducible because normally these are the structural genes again here. These are the genes that we would want active if we needed them. Well, normally they're going to be inactive. I mean, think about, how often do you give E. coli shot? There's E. coli in your belly, by the way. Okay, how often do E. coli in your belly get milk? It's not like you're drinking milk all the time. If you were a baby, well, that'd be a different story, wouldn't it? But you drank juice for breakfast. Maybe you don't have milk until lunchtime. Well, until lunchtime, this is what's going to happen with E. coli. They're going to have that repressor protein on there because it wouldn't want to expend the energy to make enzymes to break down lactose because lactose physically is not present. So that's what it means by inducible. Something has to induce this operon to work, okay? So that's what I mean by inducible operon. And well, then there's also a repressible. And in the repressible operon, it's going to be just the opposite of inducible. Repressible means that really these structural genes, they're almost always going to be transcribed. And one example is tryptophan. This is an amino acid that's required for a lot of E. coli cell functions. And so there is a regulator gene on it, and there is a repressor, but normally the repressor for this TRP operon, normally that's inactive, so the RNA polymerase can just crank right along the double helix and basically make tryptophan. It's going to be coding for production of tryptophan or different enzymes, which will be ultimately made into, well, it'd be making the mRNA. We're doing the transcription to make mRNA. There we go again, which would then be made into different proteins, and in this case would be tryptophan. And normally, this repressor is just hanging out. It's not doing anything. Well, then the question would be, well, is it possible that what if there was a lot of tryptophan? Would it need to keep on making tryptophan? And the answer seems logical, no. So what would happen if there's enough tryptophan? You got to tell Mr. or Mrs. E. coli, hey, stop making tryptophan. And so the tryptophan would fit into the repressor, and now it's activated. Okay, and that's what's interesting. The term here is genetic regulation. That's genetic regulation. Then there's another part to this. There's also called negative feedback inhibition, and that's, besides the genetic regulation, which was here, what also can happen with the tryptophan, which is, that's pretty cool, is the tryptophan can be used to inhibit the first enzyme that would be used to make tryptophan, so it can can really regulate the tryptophan levels quickly. And if you think about it, that makes sense. When you think about a prokaryote doesn't have a long lifespan, really needs to be very efficient with its cellular functions, which is why transcription and translation happen all in one shot. Here's another example of, hey, we really need to highly regulate. We can't afford to expend energy on things wastefully, so they don't. So here's a summary so far. I've got this little table here. Okay, we talked about the inducible operons and the repressible operons, and those guys are called negative control. I'm going to get a little bit more into that in a second. And when we talked about, you had those regulators, and this is kind of hard. This table is kind of tough, so I put these visuals on there. Here was the inducible operons. We talked about the lac operon. Okay, the repressor, the regulator, what it does is it inhibits the transcription. And so that means that if normally, this would be under normal operations, normally the structural genes over here, they're inactive. Okay, but with a repressible operon, which was our TRP operon, normally that repressor is inactive, and so the structural genes are normally active. Okay, so this visuals makes it a little bit easier. One thing I do want to mention is that the lac operon has, if you hear negative control over here, negative control is where an active repressor inhibits transcription. Well, there is positive control. Lac operon can do positive control. I'm not going to get into those details, but just want to throw that out there, because if you took AP, you probably learned about that one. But we are going to talk about positive control, but just in a different light. Again, our goal is to understand the GFP lab, and we need to understand positive control. And what positive control is where an activator protein is absolutely required before the RNA polymerase can attach. So right now, as it is, you can't get the RNA polymerase on here until we get an activator protein. And so what I want to do is just do a side-by-side here. Okay, this is a table from the previous slide, and nothing's changed up here. The biggest difference is, okay, what we're going to do with positive control is the active regulator promotes transcription where before it inhibited. So we want to promote transcription. All right, so if I want to promote transcription along here, well, if you're trying to promote, that would mean positive control. And so the repressor regulator, there's inactive, and the structural genes are inactive. So so you notice that's a little different. So here is positive control, and the translation only occurs if the activator protein binds to the inducer molecule. Okay, so here's my activator protein, and it would be bound to this activator binding site. And the inducer, notice that it fits right in there. It's a different protein. And so they kind of work together. And what that allows is the binding of the RNA polymerase. And again, this is positive control. Okay, the active regulator promoted transcription. That promoted the transcription. And so then it can go along and do its thing. Now, these visuals are okay. Let's get ready for the, let's talk about the Ahhh operon, because Ahhh operon was used in the plasmid for the GFP lab. Okay, so we're getting really close to understanding our GFP lab even better. Okay, so this is the Ahhh operon, and you've got the regulator gene, an activator binding site, a promoter, and the genes that the arabinose, the RA, RB, and RD. These are the genes that would be used to make proteins. One thing to note is that this arabinose operon has positive control, which would be here, but it also has regulation. And I say, dang, that's pretty cool, because what that means is it has negative control and it has positive control. Okay, so let's get into that a little bit more. Okay, let's look at the negative control first. RC, what that does is that regulator gene, which is right here, okay, that regulator gene is called RC. What that does is that produces a protein that binds to itself, and you'd think, well, that's pretty weird. It's making, the DNA can be read. You can have the polymerase make a little chunk of mRNA that would be translated into a protein RSC, and that protein would bind to itself, to the gene. That's kind of funky. Well, it's what it does. That's called self-regulation. Okay, which is pretty interesting. Kind of makes sense as we go along here. Okay, the RSC can also bind to other sites in the operon. Well, that's kind of interesting. So this protein that was made from that gene there can also go to different sites on the operon, and so it does that. And notice it's binding in the activator binding site. Okay, so right now, as it is, the RNA polymerase still can't transcribe for these different genes here. However, if we have the arabinose, which is going to be our inducer, this is our regulator. We've got a regulator here. If we have the inducer, the arabinose, so the sugar is physically present, it fits in there, and it changes the shape of this RSC right here, and that promotes RNA polymerase to say, hey, bub, come on down. And so now the RNA polymerase can do its thing and do its transcription and translation. And so now these genes, RA, RB, and RD, can be transcribed into the proteins and enzymes, which are proteins, whatever it's happens to be making here. And well, we didn't do that in our GFP lab. What we did in the GFP lab is, rather than have these genes RA, RB, RD, those were cut out, and those were replaced with a GFP gene right in here. So they cut that chunk out here with restriction enzymes and replaced it with the GFP gene. So now, with the GFP gene, in other words, we're not going to make the green fluorescent protein unless the arabinose is present. If the arabinose is present, that would change the shape of the RSC right here, which promote the binding of the RNA polymerase, and we would make our green fluorescent protein. Alright? And so now, when we come back to this guy, this is our plasmid map. Beta-lactamase, that's something that would produce basically it would produce a protein that would inhibit ampicillin killing the E. coli. The GFP, we talked where the source of that came from the jellyfish. And the RSC, now you can see why it's a little more complex than just an on-off switch. Okay, so these two really work in conjunction, the RSC and GFP. And that is all I wanted to share with you. What we'll do in class next time is we will discuss what happened with in the protein purification lab, why we're using the different buffers. All right, well, thank you very much.